All right, everyone, we're going to go ahead and get started. Thanks so much for joining Tip of the Mitt Watershed Council's Icebreaker Winter Speaker Series talk on Northern Michigan algal outbreaks. I'm Jen DeMoss, the Watershed Council's Communications Director, and I'm here to introduce our speakers. This is the first webinar in our series, and we have 5 more of these that will happen in February and March. So stay tuned till the end, and I'll put up a slide with more information about those. And you can sign up for the rest of the speaker series on the same page where you signed up for this one. I am very excited to turn it over to our guests, but first, just a few words. Number one, you'll be muted during the presentation. However, we will have time for questions at the end. You could type those into the Q&A box at the bottom of your screen, or wait until we introduce the Q&A section and use the raise hand function also at the bottom of your screen. Please don't type questions into the chat box. We're not going to be monitoring that for questions. We'll do our best to answer all of your questions depending on how much time we have left at the end. Also, we're recording this meeting, and that recording will be available later on our YouTube channel after it's edited, and we'll be sending a link out to you after the presentation for that. And then just some brief information, just in case this is the first time you've ever encountered us. We're a nonprofit membership-based organization with over 40 years of history protecting and improving water resources in Northern Michigan. We're dedicated to protecting our lakes, streams, wetlands, and groundwater through respected advocacy, education, water quality monitoring, research, and restoration actions. Currently, we have 13 staff members and hundreds of volunteers, which we're very grateful for. We've been operating in this area for quite some time, and our service area includes Emmett, Antrim, Charlevoix, and Sheboygan counties. And we're very thrilled to be protecting our region's precious water resources. I'm going to stop sharing my screen with you here. And now I can introduce our 2 speakers. We have Aaron Parker, who is a senior aquatic biologist at the Michigan Department of Environment, Great Lakes, and Energy. He's been working to track and understand harmful algal blooms in Michigan since Michigan Eagle began tracking blooms reported by citizens in 2016, which is a topic he'll be discussing today. We also have Dr. Jan Stevenson, a professor in the Department of Integrative Biology at Michigan State University. Jan has been researching the prevalence of golden brown algae within the Watershed Council's service area at Torch Lake, and he'll be discussing the characteristics and potential causes of these growing mats of golden brown algae. Aaron will be starting us off, so I'm going to turn off my video and give him the floor. Thanks so much all for joining us.
Okay, so can you see this?
Yes, we can see it. Thank you.
Okay. All right. Well, thank you for the introduction, Jen. So I have quite a bit of information to present, so I'm just going to jump right into it. So I'm going to start out just giving you some background about cyanobacteria in case you're not familiar with it. Oftentimes you will cyanobacteria is synonymous with the term harmful algal bloom. However, the term harmful algal bloom is a bit of a misnomer just because cyanobacteria, as the name implies, aren't actually true algae, but rather they are bacteria. However, they do have photosynthetic capability. They eat with other algae for nutrients and sunlight. And so because of that, though they are generally considered to be part of the algal community. They are found in diverse environments ranging from soil to seawater. However, mostly where you will find them, though, is in freshwater, which of course we have plenty of here in Michigan. A couple things about them: they are capable of regulating their buoyancy up and down in the water column in order to obtain ideal amounts of nutrients and sunlight. Some of them, if they run out of nitrogen, they can fix it out of the atmosphere. Of course, large or dense blooms, such as the one in this photo here, can be aesthetically displeasing. When they can emit strong odors, especially when they begin to senesce and decay later on in the season. And of course, the main reason, though, why the department has gotten involved with monitoring them is because they are capable of producing toxins and other irritants. So in general, where you will see these cyanobacteria blooms is when there are high amounts of nutrients in a water body, when surface conditions are calm, when water temperatures are high, Oops, didn't need to go there. So basically, right now here in January, very low probability of seeing a cyanobacteria bloom right now. However, if we're in the middle of August or September, it's 90, 95 degrees out, there's no wind, water conditions are calm, that's when you might see one of these blooms. In the past few decades, with the invasion of dreissenid mussels, zebra and quagga mussels, they have sort of turned that first bullet point on its head as far as only high nutrient water bodies experiencing these types of cyanobacteria blooms. There's plenty of evidence now that dreissenid mussels can indirectly cause cyanobacteria blooms because they'll readily feed on green algae and diatoms. However, they won't necessarily feed on cyanobacteria, especially if they're actively producing toxins. There's also evidence too that the cyanobacteria are better able to utilize nutrients from the dreissenid mussel pseudofeces, whereas other algae may not be able to utilize those nutrients as well. And then, of course, I can't talk about cyanobacteria without mentioning climate change in the Great Lakes region. And I'm not going to read off all of these figures, but suffice it to say that climate change is also happening here in the Great Lakes region. As I mentioned earlier, high temperatures contribute to cyanobacteria blooms. Longer seasons are basically making these seasons longer for us. For example, in 2020, we received our first we confirmed our first cyanobacteria bloom back in March, and we had some blooms lasting all the way into late November. Total precipitation and heavy precipitation events are also increasing in the Great Lakes region. When that occurs, when you have large rainfall events and the water isn't able to infiltrate into the soil, that's going to lead to more nutrient runoff, which can then lead to cyanobacteria. So as far as the toxins that these cyanobacteria produce, you can't tell just by looking at a bloom like this and know whether or not it is or it is not producing toxins. Pretty much the only way to do it is to perform some sort of test. And whether or not they are producing toxins is going to depend on whether they have the genotype that allows them to produce toxins. Some cyanobacteria do not have the genotype that allows them to produce toxins. Then even if they do have those genes that allow them to, they may or may not be expressing that at the time. That is, they may or may not be actively producing toxins at that time. However, if you have a population with the genes that allow them to produce toxins and they are expressing it at the time, then yes, you can have toxins in a bloom like that. So we started keeping track of just algal bloom complaints from citizens in 2013. And at that time, we only had 9 complaints. The next year in 2014, that number jumped up quite a bit, but the significant thing that happened in 2014 is the now infamous Toledo water crisis, as it's become to be known. And this basically got the attention of the governor's office at the time. Basically, they asked, they said, is this something that we should be concerned about? Obviously Lake Erie touches Michigan. How much do we really know about cyanobacteria? And the answer was, well, not a whole lot. And so we received some funding for some increased, for some new equipment. We received some sondes that have phycocyanin sensors on them. Phycocyanin is a pigment that is unique to cyanobacteria. We received rapid test kits. Our lab at the DHHS in Lansing, they received upgrades to their facilities so that they could begin testing for cyanobacterial toxins. We gave out a couple of grants to some universities, and then we also launched our bloom at michigan.gov email. And this is the email that citizens can send pictures to us if they believe that they are near a water body that is experiencing a bloom. So any of you that are watching this today, if you feel that you see a cyanobacteria bloom, you can feel free to email us there. The one thing that we do ask is that you please send us photos, because that is very helpful for us. As far as sampling, typically when we sample, we don't have a boat available to us. It's usually not a problem because oftentimes the blooms will be washed up along the shoreline anyway. When we do sample by boat, we'll take a sample in the middle of a water body and then typically several areas along the shoreline. One of the other things that we've done is if there's been a bloom, but then there's a relatively clear demarcation between the clear water that's nearby and then the scum, we'll collect a sample right in the middle of the scum and then also in the clear water nearby, and then compare toxin concentrations between those. And I'll get into that later. So like I said, we have the SONs that, so we can tell immediately whether there's phycocyanin in there, and we can tell immediately whether or not we're dealing with cyanobacteria. We will collect the sample. We'll do a rapid test strip on it, which basically tells us whether or not Microsystem is present or not. If the Microsystem is present, we will then send that to our lab for mass spec analysis. We will then communicate those results with MDAR, DHHS, us. It will then go to a local health department, and then it's up to the local health departments whether they want to post signs or issue a media release about the bloom. So what have we learned so far? First of all, we've learned that cyanobacteria comes in many different shapes and colors. So this is what you're typically going to see, this green color. However, you'll also see colors like this that are a blue or a dirty white color. This often happens when the cyanobacteria is senescing or dying. There'll often be a very foul odor associated when you start seeing these colors. We've also seen purplish colored cyanobacteria, sort of this straw or tan colored cyanobacteria. In this photo, there's some red along the edge of this bloom. And then this is just a very good photo that we received a few years ago of some very vibrant, bright blue cyanobacteria that had washed up along the shore. And this was, we actually had some citizens who were concerned that paint had been dumped on this lake with cyanobacteria. So like I said, just because you see an obvious cyanobacteria bloom doesn't necessarily mean that it's producing toxins. And sometimes just very small amounts of cyanobacteria, though, can be producing large amounts of toxins. And so this is where I'm going to start throwing numbers at you. And for reference, the EPA's recreational guidance for Microsystem is 8 micrograms per liter. So in this photo on the left, that's from a lake in Muskegon County, Mona Lake. And really in this photo, you can barely even see any cyanobacteria. It's just a very thin sheen along the water surface. However, that particular sample had 17 micrograms per liter. This photograph on the right, a small lake in Oakland County. There's an obvious cyanobacteria bloom occurring there. However, there was no Microsystem in that sample. Also, in any given lake, the cyanobacteria blooms are going to vary. They may not even happen at all during a certain year, and then certain years they may be very bad. So this Mona Lake in Muskegon County, I just looked at maximum Microsystem concentrations over years, and as you can see, there's some years where we didn't find any at all because there weren't any blooms. However, there were other years where they had very large blooms, and we had almost 500 micrograms per liter Microsystem. Also, these things move around. They're on the surface, and so in this particular lake, we saw a bloom that was on the east side of this lake. A few weeks later, it had moved down to the southern part of this lake, had a very high concentration of Microsystem then. Week later, moved up into this northeast cove of this lake, had 100 micrograms per liter. And then a couple weeks later, it had moved all the way to the west side of this lake. This was all a function of wind direction. That is why this bloom moved around. And so this is important to keep in mind, because I'll often have people say to me, they'll be like, well, I saw the bloom in front of so-and-so's house or in front of such and such business. So I think they're the ones that are contributing to the nutrients that are causing this bloom. And, you know, I have to tell them that I have literally watched blooms move across an entire lake in a matter of hours just because the wind direction changed. And so it might be in front of your neighbor's house in the morning, and then it might move and be across the lake in front of some woods in the afternoon. So location of the bloom is not going to be indicative of the nutrient source that may or may not be contributing. So earlier I mentioned how we would collect samples within the scum themselves, and then also in adjacent clear water. In this example here, I collected further away from the scum than this, but here there was almost 1,500 micrograms of Microsystem right there in the scum. But in the clear water further away, there were 4.5 micrograms per liter. Most of the time when we collect samples in the clear water, we don't find anything at all, even very close to the scums. So in this graph illustrates that. So in the scums, we found an average of around 700 micrograms per liter Microsystem. And for the ambient or clear water, you can just barely see the top of the error bar for what we see in those samples. And so this is important because if, say, you have a large lake that's 100 acres and there's one very localized bloom on one side of it, we're very confident that in the clear water, based on what we've seen, there is not going to be toxins in there. Where you're going to find most of the toxins are where it's obvious in those scums. As far as where we have seen them throughout the state, this is 2016 through 2020. There will be more once we get all the 2021 numbers finalized, but the general pattern is going to be the same. Where we see most of the blooms is in the southern half of the lower peninsula, less in the northern half of the lower peninsula, and then a handful in the upper peninsula. You'll notice that I have these lakes labeled by whether they are natural lakes, excavated, whether they have natural dams or they're reservoirs. And as you can see, over half of the lakes are either reservoirs or natural lakes with dams. And so this is noteworthy because of the 11,000 or so lakes that are greater than 5 acres in the state, only about 11% of them are these natural lakes with dams or reservoirs. So they're definitely overrepresented as far as lakes that experience these. And this is just a graph I did regressing county latitude versus number of blooms, showing that most of them occur in the southern. And then these are just some lakes in the tip of the Mitt's coverage area that have experienced these blooms. And I'm looking at the time, I realize we're getting close. So I'm gonna move through these next slides pretty fast. Comparing reservoirs, the reservoirs tend to be shallower than the other lake types. And then looking at shoreline development factor, which is I'm very sorry about my dog back there. Shoreline development factor is a measurement of the shoreline irregularity of a lake. The round lakes will have a shoreline development factor of 1. The more irregular a lake becomes, the lakes will have a higher shoreline development factor, and the reservoirs tend to have higher shoreline development factors. As far as numbers of bloom reports, those are increasing every year. The amount of cyanobacteria blooms that we're confirming each year is increasing. Looking forward, though, we have started using this data to start to list some of these water bodies as not attaining their designated use for other indigenous aquatic life. And so with the goal of this listing being that potentially a nutrient TIMDL could be developed for these water bodies, or best management practices could be funded for these water. And that is it for me now. So I am going to stop sharing so that Dr. Stevenson can his presentation.
Well, thank you so much, Aaron. That was super informative. I can see we have some questions down below, and I assure you that we'll be getting to those at the end of the presentation. And now we're going to let Jan Stevenson go ahead and share his slides.
All right, so I assume you're seeing the correct view here, yes?
Yes, we are.
Thank you. So today I want to talk about an interesting problem that I got involved with a number of years with Three Lakes Association and studying the development of these golden brown algal mats that people find aesthetically unpleasing, that also indicate a change in the lakes. That has turned out to be a bit more of a challenge to figure out what's causing them than we expect. So there are a lot of people that are involved with collecting a lot of the data that I'm going to talk about today. And I've not listed them as co-authors because there's still a lot of debate about what's actually causing these things. And I'm going to present some hypotheses and actually go down through what we've learned and how we've tested hypotheses using kind of a causal framework to evaluate those hypotheses and try to figure out the cause of this GBA, this golden brown algae that's occurring on the bottom of the lakes. Okay, so advancing slides has turned out to be more entertaining than pop. All right, so this all started around 2013 when several of us were contacted by the Three Lakes Association and talking about golden brown algae. And for the first couple of years, we just did exploratory analyses of water chemistry and the algae that were there. And then after that, we started targeting specific hypotheses and continued more in-depth study actually up through 2021. So I've just presented a report to the Three Lakes Association about a lot of this work. I really kind of upfront want to acknowledge the effort done by the Three Lakes Association, especially Becky Morris, who's chaired the Water Quality Committee for a number of years and just always been in the field, often assisted by Dean or Trish or Rick or Fred. And then Art is sometimes in the field, and sometimes he's up in a plane flying around taking these amazing aerial images of the algae in the lakes, in the Three Lakes, but also other lakes in the region as well. Also, Three Lakes Associations funded a lot of the water chemistry that's been done. And that water chemistry was done by Tim Vavrika and some other lab techs at the University of Michigan's biological station. Rex Lau and Pat Kiselak are two of my friends and colleagues, algal ecologists that are at the University of Michigan's biological station, and Shank Pan and Sherry were two people here at MSU that helped me with some of this. So this has been the work, a lot of work by a lot of people over the last 6 or 7 years. And right off the bat, we knew this was an algae that was on the bottom of the lake just from looking at it. And we generated a number of different hypotheses about what could be causing this, from zebra mussels focusing phosphorus on the bottom. And these hypotheses are kind of listed in order that they emerged in the conversation, not by order of priority. So the first idea was zebra mussels were focusing phosphorus on the bottom, and that was stimulating the algal growth. Maybe changing light conditions because the zebra mussels are pulling algae out of the water column, and that's going to increase the light to the bottom of the lake. Potentially it's runoff from big storms. It's also possible that we lost some important herbivores, grazers of algae, so nothing was stopping them from accumulating, and there's examples of that. There are issues with non-native species. In some cases, it's argued that there are non-native algal species that will invade a habitat and start causing problems. So that's another possibility. One of the things I was concerned about, this is about the time that harmful algal blooms were really hitting the news, especially in Michigan, but also biofuels and algae were just in the news everywhere. So I was a little concerned that people were just starting to notice the algae on the bottom of the lake. And it might have been there all along, but nobody really noticed it. Climate change in a warmer lake could generate this GVA. And in addition, it's possible that this is due to groundwater contamination. And if it's that, then we really want to get on that as quickly as possible to start working on solutions. From the very beginning, we kept in the back of our mind one of the key questions, which is what has changed in the lakes that could have caused this golden brown algae to develop over the time frame that we think it developed. So the early studies showed that both phosphorus and nitrogen are really low in these lakes. Both are low enough to limit algal growth, but phosphorus is substantially lower. That most of the algae that occurs in these assemblages was diatoms with very few cyanobacteria and green algae. That will change with some deeper samples that we've taken in more recently that show more cyanobacteria in some of these deeper samples, and I'll show you that in a minute. And in general, it seems like there's a shift from algae down in the sands, attached to sand grains and in the interstitial spaces among the sand grains, so down below the surface of the sand, where millions of cells per centimeter squared are typically found in these assemblages. There's been a shift from algae then on the sand grains and among the sand grains to algae on top of the sand. And pretty much the same densities, but now they're on top of the sand. So that's kind of the general conclusions early on. And so from some recent samples, I thought I'd give you an idea of what this assemblage looks like. I brought these samples back home, actually set them on a white plate, filled the white plate with water so that they would be suspended. And you can see that up in this upper left-hand panel that there's a crust, a golden brown crust that holds together on the very surface. And this is attached really tightly to a mucilaginous matrix of calcium carbonate and algae below that surface crust. And then below that, there's a lot of less tightly attached calcium carbonate that came off as it was jostled around in the plate, but which came off, all of this came off in my hand from the samples that we sampled from the bottom of the lake. As I zoom in and kind of look at things in different sections, this is the second panel to the right in the upper row is kind of a chunk of this surface mat with diatoms around the edge and some appearance of some smaller cells of algae down below. And then this mass of material up above that light is not penetrating very well. When I focus in on that in this third panel, it's apparent that these are coccoid cyanobacteria that are underlaying a golden mucilage that they've apparently secreted above the cells. So we've got cyanobacteria forming a surface crust with diatoms attached to it. When I look at the water around, they're just rafts of diatoms, these golden brown algae that have glass cell walls that are highly modal, that are moving up and down through this mat, that are here forming rafts and attaching to one another in the matrix. So now I'm going to look at this gray material in more detail. And embedded in this gray material, this calcium carbonate, these calcium carbonate granules are many, many diatoms with extensive mucilages and some filaments of cyanobacteria. But most of it are these diatoms, these golden brown diatoms that are in this matrix. So this is what composes the crust in the deeper waters that is extensive throughout the lake. So I'm going to start addressing these hypotheses, kind of working down through them fairly rapidly, show you some brief evidence that we've reduced our focus on some of these hypotheses for one reason or another, and I'll explain those reasons. So the reasons are basically associated with this whole concept about diagnosing causes that the EPA, governments around the world use to evaluate environmental problems. What's actually causing the problem? You can often get correlations between an environmental change and an effect, but you don't know it's actually the cause because you might not have measured the actual cause, the environmental change that generated that effect. So you need multiple lines of evidence, or this weight of evidence approach, for diagnosing the causes. So some of these pieces that I'm going to talk about a little bit are temporality. You need the cause to precede the effect. In an ecological relationship, we need some relationship between the environmental change and the effect. And then biological plausibility relates to, it has to make sense. So later I'm going to talk about the idea that potentially decreases in phosphorus are causing algae to accumulate in these lakes. That seems counterintuitive because the more nutrients you have, typically the more algae you get. Kind of like what Aaron was talking about, though, it's not that simple. That if cyanobacteria, if zebra mussels or dracaenid mussels are filtering the algae out of the water column, except for the cyanobacteria, then that leaves the cyanobacteria to use up all the rest of the nutrients. So you get cyanobacteria blooms even in low nutrient lakes. In this situation, it's a little more complicated that we have reproduction of the organisms that causes accumulation. And at low nutrients, you have very slow reproduction. But that doesn't mean you can't get high accumulations. If you have low rates of grazing, if there's low rates of biological disturbance and sloughing of the organisms, or those low rates of death due to bacterial and viral processes. So you can get accumulations in low nutrients, but this is an example of biological plausibility. I'm not going to talk about the experiments that I've been doing to kind of test these hypotheses where you manipulate one factor at a time, but that's another approach that we've used. All right, so some of the first evidence as we start to work on these hypotheses is we reduced the likelihood of some of these hypotheses just based on the lack of evidence. We didn't have actual evidence of changes in these things. So it's difficult to emphasize a hypothesis when there's no evidence in existing data, no evidence in the literature, or very little evidence. I don't want to say no, but it's very little likelihood that based on the evidence of a change that these things could have occurred. So we downweighted zebra mussels and the associated potential changes in light conditions. Biological plausibility is also associated with there. We don't know of effects of grazers. We don't know of algal invasive species being significant in the region, so we downweight that. With the aerial photography, we came up with a pretty good evaluation of that the starting date for these algal blooms had to be between 2010 and 2015. Art Hoadley has done a tremendous amount of work flying around the margins of these lakes, generating thousands of images that we have been using, I've been using in particular, to assess algal biomass simply by looking at how golden brown the sands are around the edges of the lake. And you can see here the same location to turn different directions along Torch Lake, where in 2010, there was no golden brown algae. And in 2015, there was a lot of golden brown algae. So we figured sometime in that time frame, it came in. So another piece of evidence that we got was from remote sensing to test the climate change hypothesis. And we used Landsat images, which give you a temperature measurement using them. And over the time period from 2000 to 2015, or I can actually see the bottom of that, but along that time period, temperature varied from around 0 to about 21 degrees diurnally with these monthly averages of temperatures. And we do not see an increase in temperature over this time period, particularly when the GBA was first observed. So we don't have temporality, and we don't have a gradient. We don't have a change in the environmental conditions. So because of that, we've reduced the likelihood that we've got algae in the news and on your mind, because we know when we have some really strong evidence that it wasn't there in 2010 and earlier. And we don't see a change in temperature of the lakes, so that is not likely a cause. Now you notice I've just kind of grayed these hypotheses out and not remove them. These are all still hypotheses that we have in our mind. So for example, zebra mussels are going to come back into a conceptual model later for what could be causing the GBA. And there are anecdotal observations that the shoals of the lake have gotten warmer. The water in the shoals of the lake have gotten warmer. So we still want to consider all of these hypotheses. We're not eliminating them, but we're reducing the likelihood that they're associated with the cause. So now I want to get into the big one associated with groundwater contamination. And the idea here is that water falls on, through rain, falls onto the surface of the land. Some of it runs off, some of it percolates down to the subsurface groundwater and through flow enters water bodies, or percolates down to the groundwater table and then enters the lake. When we have septic waste that get introduced into this rainfall and into the groundwaters, those can flow into the water. So we're concerned about septic waste getting in there, particularly phosphorus. And there's a paradigm that phosphorus does not move through groundwater, and that's pretty much been debunked. That if groundwaters become saturated with phosphorus in the right conditions, phosphorus can move through groundwaters. So we're concerned about these low phosphorus lakes having phosphorus enrichment through the groundwater and causing algal blooms. This was not very well understood around 2015 when we started this work, but recently there's been a paper by Yvonne Bonnebecanur and a variety of her colleagues, a whole host of people from around the world, talking about this relatively unstudied problem of groundwater potentially causing algal blooms in lakes. Most of that conversation is about green algae and cyanobacterial blooms, not diatoms, but some of it is about diatoms associated with nutrient enrichment. So we're thinking nutrient enrichment might be a problem. So the way we addressed that was by putting piezometers and measuring groundwater in the bottom of the lake, using piezometers in these well points and drawing water up through tubes that were buried deep in the lake set, well, 2 or 3 feet into the lake sediment, so that we were getting groundwater samples versus surface water samples. And we expected to find that groundwater would be higher in phosphorus than the surface water based on historic data. And we expect that if there's some enrichment, that this should be even higher. So in areas where we have low home densities, we're going to have really low phosphorus in the groundwater compared to surface water. In areas with higher home densities, we'd see that septic system signal of higher phosphorus concentrations. We did not observe that consistently at all. In the data that we've got over the 5 years, we are not seeing consistent associations between high phosphorus concentrations in the sediments and the groundwater being associated with changes in riparian land use. We're seeing a lot of variability in groundwater phosphorus conditions, but we can't associate it clearly with home densities. In addition, we're not finding really good associations with variability in groundwater and the amount of algae, the amount of GBA. These are results from a survey that I did with a benthic algal sampler made from Home Depot parts that scoops up algae off the bottom and has a GoPro camera taking pictures. And from the GoPro images, I was able to basically measure the amount of algae that's on the bottom. And in 2 seasons, as we go around the lake, we see an increase in algal biomass on the bottom, but we don't find good association with where there's a problem or not. So for example, in areas where we find well water and groundwater phosphorus concentrations relatively low, we'll have high GBA biomass. So, and all of these measurements of GBA biomass are well above what they were back in 2010, much higher than they were in the past. So we're not finding spatial relationships between GBA and groundwater phosphorus, and we're not finding temporal changes either. So, well, I'm sorry, yeah, this is another spatial scale where I used the aerial photographs over a number of years, and looking at that, again, we're not, with our Hoadley's aerial photographs, we're not seeing a relationship between GBA and where we're finding higher phosphorus concentrations, except in a few locations. Here's the temporal part. There was a study of groundwater that was sampled around Torch Lake in actually the sampling was done in 2005. When we compare the phosphorus concentrations in the groundwater at that time, the phosphorus concentrations that we're measuring in 2015 to 2020, they're about equal. In fact, they're a little lower now than they were back then, but probably not statistically lower. So I'd say they're about equal. So we're not seeing an increase in groundwater phosphorus concentrations during the time when the GBA developed. So we need to downplay that idea. Now we're in search of other possibilities. And one of the things that I came across in the literature was the idea that phosphorus might be changing in water. So it's actually in data, not in the literature, data in the region. One of those sets of data, so this is a decrease in phosphorus over the last 20 years in Torch Lake, Lake Bel Air, and Clam Lake that was available in the CLMP data, the historic CLMP data collected by the state. This same pattern occurs in lakes across northern Michigan. So if we go down through this, there's a list of lakes, and using Tip of the Mitt Watershed Council's monitoring data for these lakes, many of these lakes have significant decreases in phosphorus. The only ones that do not are Grand Traverse Bay, even though Little Traverse Bay does have a significant decrease, and some of the small lakes. Most of the larger lakes, particularly the largest lakes, have very strong decreases in phosphorus in these lakes. So could phosphorus be causing this problem? So we know there's a decrease in phosphorus more recently. In addition, there's some indications using paleolimnology and some work by Sherry Fritz and colleagues that was published back in 1993, using changes in diatoms and cores collected from lakes throughout Michigan, finding that if you estimate phosphorus based on diatom preferences for phosphorus, that around the mid-1900s in many of the lakes, Bel Air, Elk, Glen, not so much intermediate, there started to be a decrease in phosphorus that goes all the way back to the mid-1900s. So how could phosphorus, a decrease in phosphorus, cause increases in algae? Well, I kind of explained to you, as long as the algae grows slowly and nothing disturbs them, you can get high biomasses. There are examples of that in the Everglades with these really thick calcareous algal mats with some of the same species that I'm finding in Torch Lake and Lake Bel Air, where in very low phosphorus, that is the characteristic habitat where these thick algal mats develop. And also Didymosphenia is another example of a diatom that grows to huge biomasses, but only in low phosphorus streams. In fact, as phosphorus decreases in streams, that's when we see these Didymosphenia blooms occur. What could cause a decrease in phosphorus? We're not quite sure, but a couple of possibilities are zebra mussels over the last 20 or 30 years could have decreased phosphorus concentrations. For longer periods of time, it's been shown in Sweden that atmospheric deposition of nitrogen can reduce phosphorus in lakes. So in Scandinavia, there's a really good atmospheric deposition gradient from, and they've documented 20 years ago that nitrogen deposition can decrease phosphorus in lakes. And historically, back in the 1980s and 1990s, before we started an atmospheric deposition program to reduce nitrogen dispersal into the air, there were high nitrogen deposition rates in northern Michigan. So phosphorus decrease in the water seems like a hypothesis that is consistent with the evidence. It's biologically plausible. It's temporally possible because perhaps we have a tipping point where now the GBA blooms, which is the same thing that happens in the Everglades and with Didymosphenia. You get to a low phosphorus concentration, and there's a tipping point in that concentration where suddenly the algae can grow. So we got a shift in species composition of what happens in the mats. So this is a plausible hypothesis. We've got evidence for it. It's really novel. It's really different. I'm not sure that this is it. We're still looking at groundwater. We're still looking at the other factors. We need a lot more work to be certain about what's causing the GBA. And then just a shout out for, we need more regular monitoring assessment of these lakes for a wider diversity of conditions to really evaluate when environmental changes happen and what those causes might be. There are a number of things here that we just do not have historic evidence for testing hypotheses about why a sudden change in a lake might occur. Sampling every 5 to 10 years a broader diversity of parameters would not cost that much and would be highly beneficial for understanding future changes in these lakes. Thanks for your time.
Jan, thank you so much for that. That was great. We're going to start taking questions. I'm going to start with the chat. If I do call on you and you have your hand raised, please remember to just say which person that you have a question for. We've got quite a lot of questions. We're going to do our best here. I am going to start off with a question for Aaron. Are there any steps that can be taken against an existing algal bloom and or microsystem toxicity?
Yeah, so what do you do when you have, when there's a bloom there? I mean, there's things that can be done once it's there. I guess reactive approaches. And that's, I mean, that's going to be up to the individual lake association. And typically the most common reaction that the lake associations do is they'll hire a contractor to spray typically copper sulfate on it. But that's a decision that's made at the association level. It's not something that the state pays for or anything like that.
All right. I have a question for Jan. Why did you dismiss light conditions? Conventional thinking says that mussels have increased light levels at the bottom of deeper lakes.
So first, I haven't eliminated that hypothesis, but I've downweighted it because historic aerial photographs show light penetration on these shoals that's sufficient for algae to grow. So if you look at first principles of algal responses to light, algal growth rates saturate at very low light levels, particularly benthic algae. So just based on biological plausibility and the expected change in light that I expect in a really clear lake with zebra mussels, which we haven't seen, I've downweighted light.
Another question for Aaron. What are the human health or animal health data in terms of death or illness for HABs in Michigan?
Yeah, so I don't have those data readily available. So those are my colleagues at DHHS keep track of those. And then my colleagues at MDARD keep track of the animal data. I can say that we have had confirmed canine fatalities here in the state. Participated in a pretty thorough investigation of a private pond where several canines had died, and we did find anatoxin in the algae samples there, which anatoxin is a neurotoxin, and the descriptions of how the dogs died matched up with anatoxin toxicity. But I can say though that we've had confirmed canine deaths in the state, and we have had confirmed human illnesses as well in the state.
Okay. And just a reminder too, to put your questions into the Q&A. The chat is not being monitored. This is a question for Jan. What is the negative impact of golden brown algae, if any?
I think it's mostly aesthetic as far as we know, although you can imagine that it's affecting the benthic invertebrates on the bottom of the lake and biodiversity. But we really don't know. Just aesthetics. At this point, aesthetics is enough.
Okay. I have another question for Aaron. This is about a graphic that you showed. Did you say that lakes with dams and reservoirs are overrepresented on that map? I just wanted to make sure I heard it correctly.
Yeah, I saw that question too. I'm glad you asked that. So when I say overrepresented, so that in that context, so of the over 11,000 lakes in the state that are greater than 5 acres in size, only about 11% of those are either reservoirs or natural lakes with dams. And so then you would expect then that, well, only 11% of the lakes that I see with cyanobacteria blooms are either reservoirs or lakes, natural lakes with dams. So when I say overrepresented though, I say overrepresented as far as lakes that experience cyanobacteria blooms. So the fact that over half of the lakes that we've encountered that have had these blooms have been either reservoirs or natural lakes with dams is what's noteworthy, because it's not what you expect, because they're definitely the minority of the lakes in the state.
Here's another question for Jan. What about, and I'm sorry, I don't know these acronyms. What about the 2012 NLA data, which showed significant total phosphorus increase in the nation's lakes?
So that's a generality for the lakes of the whole nation. There's also a paper out that I've got to look back up that I was arguing with at the time, where there is a decrease in phosphorus in a number of lakes. And so figuring out that puzzle is a bit of a challenge. There are some lakes where it's decreasing, and actually in northern Michigan, we're seeing some lakes where there's no change.
Mm-hmm.
But there are some lakes that seem to be potentially sensitive to it. Ecologically, you can imagine why that would be the case. Deeper lakes, the algae that settle into the bottom, the phosphorus doesn't come back up into the aluminum. So there are going to be some lakes that will respond. There'll be some regions. Michigan and the Midwest is a hot spot for nitrogen deposition, and yeah. So, but again, this is just, it's coming up with an explanation that's consistent with our observations, with the evidence, and biologically plausible. This is kind of a new idea, and although some people in Scandinavia have been looking at the effect of nitrogen deposition on changes in phosphorus and on benthic algae. So it's not totally new.
Okay, great. I'm going to ask these two questions together for you, Aaron, because they're both about the microsystem toxin. One is, does the microsystem toxin just naturally degrade? And can you identify the kits used for quick, in parentheses, dipstick testing for toxins?
Yeah, as far as I can tell, Eurofins Abraxas is the only company that makes those rapid test kits. I actually know that because they came up for bid last year, and so I had to do some research into anybody else who uses them, because it looked like we were unfairly choosing them and not anybody else. And I said, I've looked everywhere, and they seem to be the only ones who make that. And then yes, microsystem degradation. So from what I can tell, they do seem to rapidly degrade in the environment. So once they are extracellular, so for a while there was always concern about what happens after the cyanobacteria dies and the microsystem toxins are released into the water. And there was a paper that came out a while ago that basically found in a lab setting that, yeah, these microsystem toxins last quite a while. The problem, though, is that was in a lab setting. In the field, it's the evidence is starting to point to, and based on what I've observed, I just do not find them in the clear water. And I have sampled spots literally right next to dying cyanobacteria that was just sprayed by an algicide applicator. Did not find any. There's a lot of evidence that it photodegrades, so the sunlight breaks it down, and also there's bacteria that will consume it. So that's what and the other thing too is, I mean, part of our sampling protocol is when we collect these samples, I mean, we have to keep them on ice. We have to keep them in a dark cooler, because if we leave them sitting out in the sunlight, then we're not going to get an accurate sample.
Y'all, you have such great questions, and we have totally run out of time for them. I want to thank you two presenters for doing such a great job covering such fascinating topics. You've really caused a lot of discussion. I'm going to be sending out this recording. There'll be more instructions about contacting us, and we could probably pass on questions to these presenters, but I just want to thank you so much for your time and for taking it out of your day to spend it with us. I'm going to share very quickly a slide. Oh my gosh, if it will pop up. Sorry, PowerPoint's been doing terrible things to us all day. But basically, I just wanted to share with you a slide about the rest of our presentations. It's not going to show up. Well, basically, I just want to let you know on our website, www.watershedcouncil.org, if you go to our events, you can look and see the rest of the icebreakers that we have for you coming up. Our next one is on loons and avian botulism. We have a really great one coming up on wild rice. We have one with Brian Kosminski of True North Trout, sharing his experiences as fly fishing and some fly fishing tips. So there's just a wealth of information coming to you in February and March. Also, we're a member-based organization. And so if you care to become a member, if you'd like to donate to our organization, the link for donating is also at www.watershedcouncil.org. And as I mentioned, I'm going to be sending out an email. It's going to have a link to the YouTube link for this presentation. So look for that in your emails coming up within about a week. Thank you everyone for attending and for presenting. We really appreciate your time.
Thanks, Jen.
Thank you.
All right. Have a good day, everyone.
You too.